In the crushing darkness, 3 miles beneath the waves, something bioluminescent drifts past your viewport. It has teeth like daggers, skin transparent as glass, and eyes that dwarf its entire body. This is real, and tonight, as you drift into sleep, we're descending into the abyss where the laws of biology bend, break, and birth nightmares more alien than anything Hollywood could imagine. Welcome to the deep sea, where every creature is a masterpiece of the impossible. As we leave behind the warm, sunlit waters where dolphins play and coral reefs flourish, our descent carries us into a realm that exists in perpetual dusk. The transition is gradual yet profound, like watching the sun set in slow motion over the course of hundreds of feet. At around 200 meters below the surface, we cross an invisible threshold into the mesopelagic zone, a vast layer of ocean that oceanographers have dubbed the Twilight Zone. Here, suspended between the world of light above and the absolute darkness below, only the faintest traces of sunlight manage to penetrate perhaps 1% of the brilliant rays that dance across the surface waves. This dim, blue-gray illumination creates an environment unlike any other on Earth, a place where the fundamental rules of survival have been rewritten by evolution's patient hand. In this shadowy realm, visibility becomes a deadly liability and invisibility the ultimate prize. The creatures that inhabit these depths have faced a singular challenge for millions of years: how to hunt, hide, and survive when there's just enough light to be seen by predators, but not enough to see clearly. The solution they've evolved is nothing short of miraculous. Rather than relying on pigmentation or physical camouflage like their shallow water cousins, the inhabitants of the Twilight Zone have mastered something far more sophisticated. They've learned to become light itself. Bioluminescence, the production of light through chemical reactions within living tissue, represents one of nature's most elegant solutions to an existential problem. It's a technology that predates human civilization by hundreds of millions of years, yet it's so advanced that we're still studying it in laboratories today, trying to unlock its secrets for medical imaging and biotechnology. Consider the hatchetfish, a creature that looks as though it was designed by an artist obsessed with geometry and light. Its body is impossibly thin when viewed from the front, compressed like a silver blade, with large, tubular eyes that point perpetually upward toward the faint glow above. But it's the underside of this remarkable fish that reveals its genius. Arranged in precise rows along its belly are photophores, specialized light-producing organs that function like biological LEDs. These aren't simple on-off switches. They're sophisticated instruments capable of detecting the ambient light filtering down from above and matching it exactly. When a predator lurks below the hatchetfish, looking up toward the slightly brighter surface, it sees nothing. The fish has erased its own shadow, blending seamlessly with the background light. This technique, called counter-illumination, requires constant adjustment as the fish moves through different depths, and as the surface light changes throughout the day. It's active camouflage of the highest order, a technology that defense contractors would pay billions to replicate. Yet here it operates automatically in a fish smaller than your hand. The hatchetfish is far from alone in its mastery of bioluminescence. Throughout the Twilight Zone, countless species have evolved their own variation on this theme, each adapted to their specific ecological niche. Some use bioluminescence not for camouflage but for communication, flashing species-specific patterns to attract mates in the gloom and underwater Morse code written in living light. Others employ it as a weapon, either to startle predators with sudden bursts of brilliance or to illuminate their prey, making it easier to strike. The diversity of bioluminescent strategies in this zone is staggering, a testament to the millions of years these creatures have had to perfect their craft. Scientists estimate that up to 90% of the animals in the mesopelagic zone can produce light in some form, making the Twilight Zone perhaps the most luminous ecosystem on the planet, despite existing in near darkness. But bioluminescence is only one aspect of the extraordinary adaptations found here. The Twilight Zone is also home to one of the planet's greatest spectacles, though it occurs far from human eyes and wasn't discovered until relatively recently. Every single evening, As the sun sets above the waves, something remarkable begins to happen in the depths. Creatures that have spent the daylight hours hiding in the dimness of the mesopelagic zone begin to stir. Fish, squid, jellyfish, and countless species of zooplankton start an upward journey, rising through hundreds of meters of water toward the productive surface layers where phytoplankton and small prey are abundant. This mass movement involves billions upon billions of individual animals, representing more biomass than all the wild land animals on Earth combined. It is, without exaggeration, the largest migration of life on our planet, and it happens every single night in oceans across the globe. This phenomenon, known as Dale vertical migration, follows a precise rhythm tied to the day-night cycle. As darkness falls and the risk of visual predators diminishes, the creatures ascend to feast on the bounty of the surface waters. They spend the night feeding, converting the ocean's primary productivity into their own flesh. Then, as dawn approaches and light begins to filter down through the water, they reverse course. Down they go, back into the protective dimness of the Twilight Zone, where they'll spend the daylight hours digesting their meals, growing, reproducing, and waiting for the safety of darkness to return. This daily journey isn't just a curiosity for marine biologists. It's a critical component of Earth's carbon cycle. As these animals feed at the surface, and then descend to digest and excrete waste in the depths, they transport vast amounts of carbon from the atmosphere down into the deep ocean, effectively acting as a biological carbon pump that helps regulate our planet's climate. The scale of this migration is difficult to comprehend. Imagine standing on the deck of a research vessel in the middle of the ocean at twilight, with an echo sounder pinging into the depths below. During the day, the sonar shows distinct layers in the water column, but as night falls, the screen lights up with movement. What oceanographers call the deep scattering layer begins to rise, appearing on monitors as a dense band of reflected sound that ascends through the water like a living blanket being drawn upward toward the surface. This layer is composed of countless organisms, so numerous that they can interfere with submarine navigation and were once mistaken for the seafloor itself by early sonar operators. The biomass involved is staggering. Recent estimates suggest there may be more than 1 billion metric tons of fish in the mesopelagic zone alone, make it potentially the largest fishery on Earth, though one that remains largely unexploited due to the technical challenges and energy costs of fishing at such depths. The creatures participate in this nightly migration have evolved remarkable physiological adaptations to handle the pressure changes involved in traveling hundreds of meters up and down through the water column twice a day. Many possess specialized swim bladders that can adjust their volume or are filled with lipids rather than gas, allowing them to change depth without the risk of barotrauma that would affect animals adapted to a single depth. Their metabolism has been fine-tuned over eons to balance the energy gained from feeding against the energy expended in making the journey, a calculation that must favor the trip or evolution would have selected for species that simply stayed put. Among the migrants are species you would recognize only from deep sea photography or nature documentaries. Lanternfish with their rows of bioluminescent photophores glowing like the windows of tiny underwater skyscrapers. Bristlemouths so numerous they may be the most abundant vertebrate on Earth yet remain virtually unknown to the general public. Various species of squid and octopus that pulse through the water column with liquid grace, and jellyfish of incredible diversity, some trailing tentacles dozens of feet long, others barely larger than a fingernail. Each species has its own preferred depth range, its own timing for the migration, and its own role in the complex food web that sustains life in this dim realm. The Twilight Zone also serves as a refuge for larger predators that hunt the migrants. Swordfish and tuna are known to dive into these depths in pursuit of prey, their streamlined bodies and powerful muscles allowing them to make rapid descents and ascents. Sperm whales, those giants of the deep, regularly plunge into the mesopelagic zone and beyond, holding their breath for up to 90 minutes as they hunt for squid in the darkness. For these air-breathing visitors, the Twilight Zone represents a pantry stocked with high-energy prey, where the physiological stress of deep diving and the crushing pressures involved As our imaginary descent continues through this zone, the light continues to fade until we reach the lower boundary, around 1 meters deep, where the last photons of sunlight are finally absorbed and scattered into nothingness. Here, at the edge between twilight and eternal night, we find ourselves at another threshold. Below us lies the midnight zone, the bathypelagic realm, where bioluminescence is no longer about matching background light, but becomes the only light that exists. The twilight zone above us, with its dim blue glow and its rivers of migrate life, represents a middle ground, a place neither fully illuminated nor completely dark, where evolution has produced some of the ocean's most innovative solutions to the eternal problems of survival. It's a reminder that the ocean is not simply a uniform mass of water, but rather a series of distinct vertical habitats, each with its own character, its own inhabitants, and its own secrets waiting to be discovered by those willing to descend into the depths, in the perpetual darkness of the deep ocean, where sunlight has not penetrated for hundreds of millions of years. Life has painted the void with its own constellation of living stars. More than 90% of the creatures dwelling in these lightless depths possess the extraordinary ability to produce their own illumination through bioluminescence, a biochemical process that transforms chemical energy directly into light with almost perfect efficiency. This widespread mastery of light production represents one of evolution's most successful innovations, arising independently in dozens of different lineages over the eons. While bioluminescence exists in some terrestrial creatures like fireflies and glowworms, nowhere on Earth has it been embraced with such enthusiasm and diversity as in the deep sea, where it has evolved into a sophisticated language spoken in flashes, glows, and pulses that convey messages of desire, deception, and danger. The chemistry underlying bioluminescence is elegantly simple in principle yet remarkably varied in execution. At its core, the process involves a light-emitting molecule called luciferin reacting with oxygen in the presence of an enzyme called luciferase. When these components combine, the luciferin is oxidized and releases energy in the form of visible light rather than heat, a reaction known as chemiluminescence. Different organisms have evolved different types of luciferin and luciferase, suggesting that bioluminescence has been invented and reinvented many times throughout evolutionary history. Some deep-sea creatures manufacture their own light-producing chemicals internally, synthesizing the necessary compounds through their own metabolic processes. Others have taken a different approach, forming symbiotic relationships with bioluminescent bacteria that they culture in specialized organs called photophores. These bacterial partners produce light continuously, and their hosts have evolved sophisticated mechanisms to control when and how much light is displayed, using shutters, filters, and reflectors made of biological tissue to modulate their glow. Perhaps no creature embodies the predatory application of bioluminescence more iconically than the anglerfish, a family of deep-sea hunters that have transformed part of their own anatomy into a living fishing rod. Female anglerfish possess a modified dorsal spine called an esca that extends forward above their cavernous mouths, dangling in the water like a luminous lure. This appendage houses colonies of bioluminescent bacteria that the anglerfish cultivates carefully, providing them with oxygen-rich blood and nutrients in exchange for their constant glow. The light produced by these bacterial partners creates an irresistible beacon in the darkness, mimicking the appearance of small prey organisms or perhaps simply representing a mysterious, glowing object worth investigating in an environment where anything edible is precious. When a curious fish or a squid approaches to investigate this strange light, it ventures unknowingly into strike range of the anglerfish's formidable jaws. In a burst of speed surprising for such an ungainly-looking creature, the anglerfish lunges forward, its mouth opening impossibly wide to engulf prey sometimes as large as itself. Different species of anglerfish have evolved distinct esca shapes and lighting patterns, each adapted to attract their preferred prey. Some possess simple bulbous lures that glow steadily, while others have elaborate, branching structures that dangle multiple points of light. creating a more complex visual display. Certain species can even manipulate the intensity of their lure's glow, creating a pulsing or flickering effect that may prove even more enticing to potential victims. This diversity suggests that prey species can learn to recognize and avoid certain lure types, driving an evolutionary arms race between anglerfish innovation and prey wariness. The male anglerfish, incidentally, has taken a different evolutionary path entirely. Much smaller than females and lacking the iconic lure, males of many species have evolved into parasitic mates that attach permanently to females, fusing their tissues and circulatory systems in one of nature's most extreme examples of sexual dimorphism and reproductive strategy. While the anglerfish uses bioluminescence to attract prey toward danger, other creatures employ light for precisely the opposite purpose, to repel predators or facilitate escape. The vampire squid, despite its ominous name, is actually a relatively gentle creature that feeds on marine snow, the constant rain of organic particles that drifts down through the water column. When threatened by predators, however, this 8-armed cephalopod deploys one of the ocean's most spectacular defensive light shows. It can avert its arms inside out, revealing dozens of bioluminescent photophores that suddenly erupt in brilliant blue light. The squid waves these glowing arms wildly while simultaneously releasing clouds of bioluminescent mucus rather than the ink employed by its shallow water relatives. This creates a confusing explosion of light in multiple directions, a strobe-like display that overwhelms the predator's visual system and disguises the squid's escape trajectory. While the predator attempts to make sense of this sudden sensory assault, trying to distinguish the actual prey from the expanding cloud of glowing decoy material, the vampire squid makes its retreat into the safety of deeper darkness. The defensive bioluminescent employed by the vampire squid represents just one strategy in an extensive playbook. Some deep-sea creatures respond to threats by producing startling flashes meant to surprise and momentarily blind predators, buying precious seconds for escape. Others use bioluminescence in what scientists call a burgle air alarm strategy. When attacked by a predator, they produce brilliant displays that attract even larger predators to the scene, essentially calling for help from the enemy of their enemy. The small, threatened creature hopes that the arrival of this larger predator will either drive off its attacker or result in the attacker itself becoming prey, allowing the original victim to escape in the chaos. This strategy has been observed in various species of dinoflagellates, small shrimp, and jellyfish, all of which can suddenly light up when disturbed, creating a glowing target that larger predators can home in on. Among the most cunning applications of bioluminescence is the strategy employed by the cookie-cutter shark, a small but remarkably successful predator that has turned the principles of counter-illumination on their head. This shark, typically less than 2 feet long, has a belly covered in photophores that glow with a soft blue-green light, matching the faint illumination filtering down from the distant surface above. This should make the shark nearly invisible when viewed from below, just like the hatchetfish and countless other species using counter-illumination as camouflage. However, the cookie-cutter shark has evolved a devious modification to this standard strategy: a small, dark patch on its throat that doesn't produce light. When a larger predator looks up from below, it doesn't see an invisible shark blending with the background light. Instead, it sees what appears to be a small fish silhouetted against the dim glow above, the dark patch mimicking the profile of prey. This illusion proves irresistible to larger fish, sharks, whales, and dolphins, all of which approach expecting an easy meal. But as the predator draws near and opens its mouth to engulf what it believes is a small, helpless fish, the cookie-cutter shark strikes with astonishing speed. It latches onto its would-be predator with specialized sucker-like lips, then rotates its body while sawing with its lower teeth, which are among the largest proportional to body size of any shark. In seconds, the cookie-cutter removes a perfectly circular plug of flesh from the victim, leaving a crater-like wound that gives the shark its common name. The shark then releases its grip and vanishes back into the darkness, leaving the much larger animal wounded and confused. Cookie-cutter shark wounds have been found on everything from great white sharks to submarines. Their distinctive circular scars, testament to the effectiveness of this audacious hunting strategy. The shark has essentially weaponized the very camouflage strategy that most deep-sea creatures use for protection, turning it into a lure that draws prey close enough to attack. Beyond predation and defense, bioluminescence serve crucial roles in one of life's other fundamental imperatives, reproduction. In the vast darkness of the deep ocean, finding a mate of the correct species presents enormous challenges. Visual cues that work in shallow, sunlit waters are useless here, and chemical signals diffuse slowly and unpredictably in the currents. Many deep-sea species have evolved species-specific bioluminescent displays that function as mating signals, allowing individuals to identify suitable partners across the void. These displays can be remarkably complex, involving precise patterns of light organs that flash in particular sequences, much like the courtship displays of fireflies on summer evenings. Some species of deep-sea squid possess elaborate light organs around their eyes or along their bodies that flash in patterns unique to their species, ensuring that mating efforts are directed toward genetically compatible partners rather than wasted on similar-looking but reproductively incompatible relatives. The hatchetfish, already discussed for its counter-illumination camouflage, also uses bioluminescence for mate recognition. Different species within the hatchetfish family can be distinguished by the precise arrangement and characteristics of their photophores, and these patterns likely play a role in helping individuals identify potential mates of their own species. Similarly, many species of lanternfish, those abundant migrants of the Twilight Zone, possess distinct photophore patterns that vary between species and sometimes between sexes within the same species. Males and females may have differently arranged light organs, allowing them to recognize each other in the darkness and coordinate their brief, crucial encounters for spawning. Some deep-sea creatures have pushed the complexity of bioluminescent communication even further, developing what appear to be true visual languages. Certain species of deep-sea shrimp have been observed producing rapid sequences of flashes when encountering conspecifics, suggesting information exchange beyond simple species recognition. While we cannot yet decode what messages these flashes might convey, the patterns are consistent enough to suggest meaning, perhaps signals of aggression, submission, or reproductive readiness. The study of these potential languages remains in its infancy, hampered by the difficulty of observing the creatures in their natural habitat and the challenge of interpreting behaviors that evolved in an environment so foreign to our own terrestrial experience. The diversity of bioluminescent organs across deep-sea species demonstrates the incredible plasticity of evolution when presented with a common challenge. Some creatures possess simple patches of light-producing tissue, while others have developed organs of stunning complexity that rival the sophistication of eyes. These advanced photophores may include reflective layers to direct and amplify light, pigmented filters to modify the color of emission, and muscular shutters to control when light is displayed. Some fish have photophores equipped with lenses that can focus the emitted light in a beam, allowing them to illuminate specific areas or create searchlight-like effects. The loosejaw fish takes this even further, possessing a suborbital photophore that produces red light, unusual in the deep sea, where most bioluminescence is blue or green. Since most deep-sea creatures cannot see red light, this gives the loosejaw a private flashlight invisible to both prey and competitors, allowing it to hunt without revealing its presence. The chemical efficiency of bioluminescence is remarkable from a bioenergetic perspective. Unlike incandescent bulbs, that waste most of their energy as heat, bioluminescent reactions convert chemical energy to light with up to 95% efficiency. This near-perfect conversion is crucial for deep-sea organisms operating under severe energy constraints, where every calorie must be carefully budgeted. In an environment where food is scarce, and the next meal may be days or weeks away, the ability to produce light without wasting precious energy as heat represents a significant survival advantage. Scientists and engineers have long studied bioluminescence, hoping to understand and harness its efficiency for human applications. From more energy-efficient lighting to medical imaging techniques that could track cellular processes in real time. In the deep ocean, bioluminescence has become more than just a survival tool. It is the primary means of interaction, communication, and perception in a world without sunlight. It is both voice and vision, weapon and shield, truth and deception. The living lights that sparkle and pulse in the darkness speak a language far older than human civilization, refined by hundreds of millions of years of evolution into dialects as diverse as the species that speak them. For the creatures of the deep, light is not simply electromagnetic radiation to be passively received, as it is for us dwelling in the sun's radiance. Instead, it is something they create, control, and wield with purpose and precision, painting meaning onto the canvas of darkness with their own luminous bodies. We have now descended beyond the reach of sunlight, crossing into a realm that had existed in absolute darkness since the oceans first formed billions of years ago. Welcome to the Twilight Zone, the midnight realm of the deep ocean, stretching from approximately 1,000 meters down to 4,000 meters below the surface. Here, in this vast expanse of perpetual night, no photon of sunlight has ever penetrated, not in all the countless eons since complex life first evolved in Earth's seas. The darkness here is not merely the absence of light we experience on a moonless night on land. It is total, complete, and unbroken except for the occasional flicker of bioluminescence from the creatures that drift through this liquid space. This is darkness as a fundamental condition of existence. Darkness that has shaped every aspect of evolution for the organisms that call this zone home. The physical conditions in the bathypelagic zone are as extreme as anything found on our planet's surface. The pressure at these depths is crushing beyond human comprehension, increasing by approximately 1 atmosphere, roughly 14.7 pounds per square inch for every 10 meters of descent. At the upper boundary of this zone, pressure already exceeds 100 atmospheres, and by the time we reach the lower limits at 4,000 meters, we're experiencing pressures approaching 400 atmospheres, or nearly 6,000 pounds per square inch. To put this in perspective, this is equivalent to having a large elephant standing on every square inch of your body simultaneously. For humans without protection, death would be instantaneous and absolute. Our lungs would collapse. Our cell membranes would rupture, and the gases dissolved in our tissues would behave in catastrophic ways. Yet for the creatures adapted to these depths, this crushing pressure is simply normal, the baseline condition against which their entire biology has been calibrated. Over millions of generations, the temperature in the midnight zone hovers just a few degrees above freezing, typically between 2 and 4 degrees Celsius. Unlike the surface waters that fluctuate with seasons and weather patterns, these temperatures remain remarkably stable year-round, creating an environment of almost unchanging physical conditions. This thermal stability has persisted for geological timescales, allowing evolution to fine-tune organisms to very specific parameters without the need for adaptability to seasonal variation. The cold also affects the behavior of biological molecules, slowing metabolic reactions and allowing these creatures to operate at a much lower energetic tempo than their warm water relatives. In some ways, the entire ecosystem runs in slow motion compared to the frenetic pace of life in shallow tropical seas. Food in the bathypelagic zone is desperately scarce, arriving primarily as marine snow, the constant gentle rain of organic particles, dead organisms, fecal pellet, and aggregated debris that sinks down from the productive surface waters far above. This nutritional drizzle is thin indeed, and the creatures here have evolved remarkable adaptations to survive on what amounts to starvation rations. Many species have dramatically reduced their metabolic rates, living life in a state of near suspended animation, moving little and growing slowly. Others have abandoned the luxury of being choosy about their diet, evolving the ability to consume almost anything organic that crosses their path, no matter how large or how unusual. Among the most striking examples of this eat-anything-you-encounter strategy is the gulper eel, also known as the pelican eel, a creature so bizarre in its proportions that it seems almost cartoonish. The gulper eel's most prominent feature is its enormous mouth, which can open to angles that seem geometrically impossible, creating a gape far wider than the eel's slender body. The jaw is loosely hinged and the skull is reduced to a delicate framework that provides minimal structure, prioritizing flexibility over strength. When the gulper eel encounters prey, it can engulf organisms considerably larger than itself, its mouth ballooning outward like a grotesque net. Behind this cavernous maw lies a stomach equally extraordinary, stretchy, elastic, and capable of expanding to accommodate meals of surprising size. The gulper eel's entire body plan represents a commitment to a simple philosophy. When food is rare, you cannot afford to pass up any opportunity, regardless of whether the prey seems too large. Better to have the ability to consume a once-in-a-month windfall than to let it escape because your mouth wasn't big enough. The gulper eel's body tapers to a long, whip-like tail that comprises the majority of its length, which can reach up to 6 feet in some species. At the tip of this tail sits a bioluminescent organ that glows with a pinkish light, likely used to attract prey in the darkness. The eel's actual body mass is minimal, its tissues reduced to the bare minimum needed for survival. It drifts through the water column more than it swims, conserving every possible calorie, its great mouth gaping open as it slowly cruises through the darkness, waiting for something edible to blunder into range. Recent research suggests that gulper eels may feed primarily on small crustaceans rather than large fish, as once thought, using their enormous mouths not to swallow single large prey items but to sweep through clouds of smaller organisms, filtering them from the water. Regardless of their exact feeding strategy, they remain testament to the strange solutions evolution produces when the ordinary rules no longer apply. If the gulper eel seems alien in its construction, the barreleye fish pushes biological design into territory that seems almost science-fictional. This extraordinary creature possesses what is arguably one of the most unusual cranial structures of any vertebrate, a complete transparent skull filled with clear, jelly-like fluid, through which we can look directly at the fish's internal anatomy. The first specimens of barreleye fish brought to the surface confused scientists for decades, as the delicate transparent dome would collapse under the pressure change, and researchers examining damaged specimens couldn't understand their true structure. It wasn't until deep-sea submersibles allowed scientists to observe living barreleye fish in their natural habitat that the full marvel of their design became apparent. Inside that fluid-filled transparent dome sit the barreleye eyes, but these are not eyes as we typically envision them. Rather than being round or elliptical organs positioned on the sides of a head like most fish, the barreleye's eyes are tubular, looking rather like the twin barrels of binoculars mounted inside the fish's head. These tubular eyes point straight upward when the fish is in its typical hunting posture, allowing it to see and the waters above for the silhouettes of prey against the faint bioluminescent glow that represents the only light at these depths. The tubular design provides exceptional light-gathering ability and allows for highly acute forward vision within a narrow field of view, perfect for spotting small prey items directly overhead. For years, scientists believed this upward staring gaze was fixed, that the barreleye fish was locked into looking only upward and would have to rotate its entire body to track prey or look in other directions. This seemed problematic. How could the fish actually eat prey position above it if it couldn't redirect its gaze forward toward its mouth? The mystery was solved when researchers using remotely operated vehicles observed living barreleyes in their natural habitat and discovered that the tubular eyes can actually rotate within the transparent skull. The fish can pivot its eyes from the upward staring position to looking forward, allowing it to track prey as it maneuvers to capture it. The fluid-filled dome serves multiple purposes. It protects the delicate eyes from the stinging cells of jellyfish whose tentacles the barreleye fish is thought to steal food from. It may help focus and concentrate the minimal available light onto the retinas, and it maintains the correct optical properties for the eyes to function in the deep-sea environment. The barreleye typically hovers motionless in the water, its large, flat fins extended to stabilize its posit. Ion, its tubular eyes scanning upward for prey. The transparency of its head is so complete that you can see through to the other side, observing the eyes, brain, and other organs suspended in the clear fluid, as if viewing anatomical specimens in a jar. This transparency may serve a camouflage function as well, making the fish harder to detect in the dim environment. What appears to be the fish's eyes when viewing it from the front are actually its olfactory organs. The true eyes are the green, glowing orbs visible within the dome, their color coming from a yellow pigment that filters out the blue light most common in deep-sea bioluminescence, enhancing contrast and making prey easier to detect. These creatures, the gulper eel with its nightmarish maw and expandable stomach, the barreleye fish with its transparent skull and rotating barrel eyes represent just two examples of the extraordinary adaptations that have evolved in the bathypelagic zone. Throughout this realm swim dozens of other species that would seem equally impossible if we encountered them in any other context. Fish with teeth so large they cannot fully close their mouths, projecting outward like fangs designed to impale rather than bite. Squid with asymmetrical eyes, one enormous for looking upward into the gloom, the other small for looking downward into greater darkness. Jellyfish of incredible delicacy, their bodies more than 99% water, drifting on currents with tentacles extended like drift eds to snare anything that touches them. Many bathypelagic fish have evolved bodies that are extremely fragile and gelatinous, a stark contrast to the muscular, active fish of shallow waters. With food so scarce, maintaining a heavy, robust body would be a wasteful expenditure of energy. Instead, these fish have reduced their skeletons to minimal frameworks, their muscles to thin sheets barely sufficient for slow swimming, and their organs to the smallest configurations that can still support life. They are creatures of minimal viable construction, stripped down to the bare essentials. Their flesh is often watery and provides little nutrition, making them unappealing prey for most predators, a defense strategy based not on armor or weaponry but on simply not being worth the effort of eating. The reproductive strategies of bathypelagic fish reflect the challenges of finding mates in the vast darkness. Some species have evolved hermaphroditism, allowing any two adults that encounter each other to mate regardless of their original sex. Others release massive quantities of eggs and sperm into the water column, hoping that random chance will bring them together in the currents. Still others, like the anglerfish mentioned previously, have evolved extreme sexual dimorphism with parasitic males that permanently attach to females, ensuring that when the rare female is ready to spawn, a mate is already present. Each strategy represents an evolutionary solution to the fundamental problem of reproduction in an environment where your own species is vanishingly rare and encountered are matters of chance. The Midnight Zone also challenges our understanding of sensory perception. In the absence of sunlight, vision would seem useless, yet many bathypelagic creatures retain highly developed eyes adapted to detect the faintest bioluminescent signals. These eyes are often enormous relative to body size, with huge pupils and dense concentrations of light-sensitive rod cells in their retinas, sacrificing color vision and visual acuity for maximum light sensitivity. Other species have abandoned vision entirely, either reducing their eyes to vestigial structures or losing them altogether, relying instead on other senses like mechoreception, the ability to detect pressure waves and water movements created by nearby organisms. Some fish have lateral line systems of extraordinary sensitivity, able to detect the minute disturbances in the water created by a small crustacean moving meters away. The bathypelagic zone represents a vast habitat comprising an enormous volume of the world ocean. If we were to calculate the actual three-dimensional space available in this zone, it would dwarf all terrestrial habitats combined. Yet despite this immense volume, the biomass living here is relatively sparse compared to the teeming life of the sunlit surface. or the productive zones near hydrothermal vents. Life in the midnight zone is spread thin, distributed across enormous distances, each organism maintaining a territory measured in cubic meters or more, drifting through the darkness in near isolation. Encounters between organisms are rare events, whether those encounters represent opportunities for feeding, mating, or simply the passage of two beings sharing the void. For humans, the midnight zone remains largely unexplored, despite being one of Earth's largest habitats. The technical challenges of reaching these depths and the expense of mounting deep-sea expedition mean that we have better maps of the surface of Mars than we have of much of the bathypelagic zone. The creatures we have discovered represent what scientists believe is likely a fraction of the actual biodiversity present at these depths. Each submersible dive or deep camera deployment reveals previously unknown species, strange forms of life adapted to conditions we are only beginning to understand. The midnight zone holds secrets we have yet to discover, organisms whose adaptation might inspire new technologies, biochemistries that could revolutionize medicine, and ecological relationships that challenge our understanding of how life persists in seemingly impossible circumstances. Here, in Perpetual Night, evolution has indeed abandoned the familiar rules and written new ones entirely. In the crushing darkness of the deep ocean, the fundamental rules governing body size seem to have been suspended, rewritten by evolution. Pressures so different from those on land or in shallow waters that the results appear to defy biological logic. Here, in this alien realm, we find creatures that challenge our intuition about what size an organism should be based on its taxonomic relationships. Crustaceans that would fit comfortably in your palm in shallow water grow to the dimensions of large dogs. Squid expand to lengths that rival school buses. Meanwhile, other lineages shrink to microscopic proportions, conserving every molecule of precious energy. This phenomenon, known as deep-sea gigantism when organisms grow unusually large, and dwarfism when they become unusually small, represents one of the ocean's most perplexing biological mysteries, one that scientists are still working to fully understand even after more than a century of study. The giant squid, Architeuthis ducks, stand as perhaps the most iconic example of deep-sea gigantism and has haunted human imagination for centuries. Sailors' tales of kraken and sea monsters that could drag entire ships beneath the waves were long dismissed as fantasy until the 19th century, when bodies of enormous squid began washing ashore and appearing in the stomachs of sperm whales. These creatures can reach confirmed lengths of 43 feet from the tip of their mantle to the end of their two elongate feeding tentacles, with some historical reports suggesting even larger specimens, though these remain unverified. Their eyes are the largest in the animal kingdom, reaching diameters of up to 10 inches roughly size of dinner plates. Adaptations for gathering every possible photon of light in the darkness where they hunt. A giant squid's 8 arms are lined with hundreds of powerful suckers, each ring with sharp, tooth-like structures called sucker rings that can grip and tear flesh. The 2 longer feeding tentacles can shoot out with explosive speed to capture prey, then retract to bring the struggling victim toward the parrot-like beak. Hidden among the arms, capable of shearing through muscle and bone for most of human history, no one had seen a living giant squid in its natural habitat. These creatures inhabited depths beyond the reach of early diving technology, and the few specimens that surfaced were dead or dying, their bodies damaged by the pressure change and unable to display their true behavior. It wasn't until 2004 that Japanese researchers Tsunemi Kubodera and Kyoichi Mori obtained the first photographs of a living giant squid in the wild, and not until 2012 that the first video footage was captured of one swimming in its natural deep-sea environment. These historic recordings revealed a creature far more active and aggressive than the sluggish, energy-conserving animals scientists had theorized them to be. The footage showed the giant squid attacking the researchers' bait with remarkable speed and power, wrapping its arms around the prey and attempting to drag it away, demonstrating that these animals are formidable predators despite their enormous size. The quiz tie-in that has perplexed biologists is simply, why? Why would a squid grow to such immense proportions when its shallow water relatives remain relatively modest in size? The giant squid's closest relatives, other species in the order Ogopsida, are typically measured in inches, not feet. What evolutionary pressures in the deep sea favor such extreme gigantism? Several hypotheses have been proposed, each likely contributing part of the answer to this complex puzzle. The cold temperatures of the deep ocean, slow metabolic rates, allowing organisms to live longer and potentially grow larger over their extended lifespans. Cold water also holds more dissolved oxygen than warm water, potentially allowing for more efficient cellular respiration to support larger body masses. The reduced gravity effect in water buoyancy means that large size doesn't carry the same structural costs it would on land, where bones and muscles must support an organism's weight against gravity. Perhaps most significantly, the deep ocean lacks many of the predators that would typically prey on medium-sized animals. In the midnight zone, there are no hunting packs of fish, no fast-swimming predators that could easily take down a moderately large animal. The main predator of giant squid is the sperm whale, a mammal that must surface to breathe and therefore cannot spend all its time hunting in the depths. For a squid, growing large enough to become difficult prey for anything except the largest whales represent a significant survival advantage. Once an animal crosses a certain size threshold, its number of potential predators drops dramatically, perhaps even to zero. This release from predation pressure may allow for the evolution of gigantism in lineage that can overcome the other challenges large size presents. The Japanese spider crab, Macrochaea campheri, represents another spectacular example of deep-sea gigantism, though it inhabits the continental shelves and slopes, rather than the open ocean depths. This arthropod holds the record for the largest leg span of any arthropod on Earth, with the largest confirmed specimens reaching 12 feet from claw to claw. Despite this enormous span, the crab's body is relatively small, creating a creature that appears almost skeletal, like a walking collection of stilts with a modest carapace suspended in the middle. These crabs can weigh up to 40 pounds and are thought to live up to 100 years, slowly growing throughout their long lives in the cold waters off Japan's Pacific coast. Their spindly legs allow them to walk across soft sediments without sinking in, distributing their weight across a large area in adaptation to moving through their muddy habitat at depths between 50 and 300 meters, occasionally venturing deeper. Unlike the giant squid's active predatory lifestyle, the Japanese spider crab is a scavenger, an opportunistic feeder, using its long legs to patrol the seafloor for dead and decaying matter, small invertebrates, and algae. Its enormous size may serve primarily defensive purposes. Few predators can manage to consume such a large, hard-shelled creature, and the crab's reach with its long limbs means it can detect and respond to threats from a considerable distance. The crab's longevity also plays a role in its size, with up to a century to grow and relatively few natural predators once it reaches adult size. Individual crabs can attain dimensions impossible for their shorter-lived relatives. Perhaps no example of deep-sea gigantism is quite as startling to observers as the giant isopods, genus Bathynomus, which belong to the same order of crustaceans as the familiar pill bugs or roly-polies that children find under garden rocks, where terrestrial pill bugs typically measure half an inch to an inch in length. Giant isopods can reach lengths of 16 inches and weights exceeding 3 pounds, making them comparable in size to a football or small cat. These armored scavengers patrol the deep ocean floor on 7 pairs of legs, their segmented exoskeleton allowing them to curl into a ball when threatened. Just like their tiny terrestrial cousins, giant isopods feed primarily on dead whales, fish, and squid that sink to the ocean floor. Using their powerful mandibles to tear into decaying flesh, they are capable of surviving for years without food, their metabolism slowing to barely detectable levels during periods of famine, engorging themselves when a food fall appears, consuming so much that they can barely move. The size difference between terrestrial pill bugs and giant isopods is so extreme that it challenges our understanding of the constraints on body size in crustaceans. On land, arthropods are limited in size by the efficiency of their respiratory systems, which rely on passive diffusion through tubes called tracheae rather than active circulation via a circulatory system. In water, however, oxygen can be extracted directly from the surrounding medium through gills, allowing for much more efficient gas exchange. The cold, oxygen-rich waters of the deep sea may remove one of the key constraints limiting arthropod size, allowing isopods to reach dimensions impossible for their air-breathing relatives. Additionally, the lack of terrestrial predators like birds and mammals, combined with the absence of competition from vertebrate scavengers, may have opened an ecological niche that favored larger body size in these deep-sea scavengers. Yet deep-sea gigantism tells only half the story. For every lineage that has evolved gigantic forms in the abyss, there are others that have moved in precisely the opposite direction, shrinking to minimize their energy requirements in this food-poor environment. Some species of deep-sea fish are mature and reproductively active at body lengths of less than an inch, representing some of the smallest vertebrates known to science. Certain anglerfish species have males that measure only a fraction of an inch in length, though these extreme dwarfs are parasitic on their much larger mates and perhaps shouldn't be considered independently viable organisms. Many deep-sea crustaceans, worms, and other invertebrates have evolved reduced body sizes compared to their shallow water relatives, the opposite trend. From the giant isopods and spider crabs, this dwarfism represents an alternative evolutionary strategy for coping with the same extreme environment. In a habitat where food arrives sporadically and unpredictably, where the next meal might be days, weeks, or even months away, maintaining a small body requires fewer calories. A small organism has a lower absolute metabolic demand, can survive on smaller prey items, and can reproduce with less energetic investment. For species that cannot adopt the sit-and-wait or scavenging strategies of larger predators, remaining small and reducing their metabolic overhead to the absolute minimum may represent the only viable survival strategy. These miniaturized species often have simplified anatomies, reduced organ systems, an extremely slow growth rate, representing biology stripped down to its bare essentials. The coexistence of both gigantism and dwarfism in the deep sea suggests that there is no single optimal body size for this environment, but rather multiple viable strategies depending on an organism's ecology, phylogenetic constraints, and specific adaptation. The deep ocean is not a uniform habitat but rather a complex mosaic of different niches: open water, seafloor, hydrothermal vents, cold sea, whale falls, and more, each potentially favoring different body size strategies. In the water column, where prey is sparse and scattered, large size might allow for more efficient cruising and better ability to capture widely dispersed prey. On the seafloor, where food falls are rare but substantial, large size allows an organism to gorge on a whale fall and then survive for extended periods without eating. For smaller, more specialized feeders, dwarfism allows survival on the constant trickle of marine snow and small prey items. Temperature appears to play a crucial role in both phenomena through a principle known as the temperature size rule, which predicts that ectothermic organisms raised in colder temperatures will grow to larger adult sizes than those in warmer conditions. The mechanisms behind this rule are still debated, but may involve the effects of temperature on cellular processes, the efficiency of oxygen delivery to tissues, and the timing of developmental milestones relative to growth rates. In the consistently cold waters of the deep sea, organisms experience temperatures that rarely rise above a few degrees Celsius, potentially biasing them toward larger sizes across many different lineages. Oxygen availability also correlates with body size in aquatic organisms. Larger animals require more oxygen to support their greater mass of metabolic active tissue, and oxygen delivery becomes increasingly challenging as body size increases. eventually hitting hard physiological limits. However, cold water can hold significantly more dissolved oxygen than warm water at 4 degrees Celsius. Water can hold nearly twice as much dissolved oxygen as water at 25 degrees Celsius. The deep ocean's combination of cold temperature and relatively high oxygen concentrations may allow organisms to overcome some of the respiratory constraints that limit body size in warmer, oxygen-poor waters. This might explain why some of the largest arthropods on Earth are found in cold, well-oxygenated deep-sea habitats rather than in tropical, shallow waters. Predation pressure, or more accurately the release from predation pressure, likely represents another crucial factor. In shallow tropical reefs, a vast array of predators ensures that most animals are consumed before they can reach large size. Fast-swimming fish, octopuses, seabirds, marine mammals, and countless other hunters create an environment where staying small and hiding is often the only viable defensive strategy. In the deep sea, the predator community is far less diverse and far less dense. There are fewer hunters per unit volume, and many of those that exist are highly specialized. An organism that can grow large enough to exceed the gape size of most deep-sea predators might find itself effectively invulnerable to predation, allowing it to reach sizes impossible in more predator-rich environments. The phenomenon of deep-sea gigantism extends beyond the most famous examples to include many other lineages. Giant sea spiders with leg spans exceeding 2 feet patrol the Antarctic seafloor. Supergiant amphipods reaching lengths of nearly 12 inches have been discovered in the Hadal Trenches. Even single-celled organisms reach extraordinary sizes in the deep sea, with some xenophyophores amoeba-like protists growing to 4 inches in diameter, making them among the largest single-celled organisms known. Each of these represents an independent evolutionary experiment in gigantism, suggesting that whatever factors promote large size in the deep sea operate broadly across many different types of organisms. The study of deep-sea gigantism and dwarfism continues to reveal new insights about the limits of body size and the factors that control organismal scaling. These extreme examples help scientists understand the constraints and opportunities that govern body size evolution more generally, illuminating principles that apply to life throughout Earth's oceans and potentially on other worlds. As we continue to explore the deep ocean with improving technology, we discover new examples of both extreme size increase and extreme miniaturization, each one adding another piece to the puzzle of why size matters, and when the normal rules no longer apply. Descending further into the abyss, we pass through 4,000 meters and enter a realm so vast and so utterly devoid of features that it challenges the human capacity to comprehend emptiness. This is the Abyssal Plain, a landscape that covers more than half of our entire planet's surface, making it the single largest habitat on Earth. Yet for all its geographical dominance, it remains one of the least explored and least understood environments in our world, stretching across the ocean basins in vast expanses that can extend for thousands of miles without interruption. The Abyssal Plains are flatter than any terrestrial desert, flatter than the Great Plains of North America, flatter even than the most featureless ice sheets of Antarctica. The seafloor here lies blanketed under thick layers of sediment, the accumulated dust of ages composed of the remains of countless microscopic organisms that lived and died in the waters above, slowly drifting downward over millennia to build deposits that can be hundreds of feet thick. The darkness here is absolute and eternal, unchanged since these plains first formed hundreds of millions of years ago. No variation in light marks the passage of days or seasons. The temperature hovers just above freezing, typically between 2 and 3 degrees Celsius, and has remained essentially constant for geological timescales. The pressure at these depths exceeds 500 atmospheres, or roughly 8,000 pounds per square inch, enough to instantly crush any air-filled cavity and compact human remains into a space no larger than a briefcase. Yet this is not the crushing pressure's most profound effect. Rather, it is the way it shapes every molecule, every cellular process, every biochemical reaction in the organisms that live here. Proteins must fold differently. Membranes must maintain flexibility at different composition, and enzymes must function under conditions that would denature and destroy the biomolecule that power life in shallow waters. The most defining characteristic of the Abyssal Plains, however, is not the darkness, nor the cold, nor even the crushing pressure. It is hunger. Food scarcity on the Abyssal Plains represents one of the most extreme nutritional challenges faced by any ecosystem on Earth. The primary source of nutrition is marine snow, that gentle, constant rain of organic particles falling from the productive waters thousands of meters above. By the time this material reaches the abyssal seafloor, it has been picked over by creatures at every depth along its descent. The more nutritious components have been consumed, the more easily digestible materials have been broken down by bacteria, and what remains is largely the refractory, difficult to metabolize remnants, the rinds and husks of the ocean's productivity. Scientists have calculated that less than 5% of the organic matter produced in surface waters actually reaches the abyssal plains, and of that small fraction, much of it is so degraded as to provide minimal nutritional value. In this environment of perpetual famine, natural selection has favored organisms capable of extraordinary efficiency, creatures that can extract every possible calorie from their environment and survive on rations that would starve their shallow water relatives within days. The fang-toothed fish, Anoplogaster cornuta, represents perhaps the most visually striking adaptation to this food-scarce existence. This small fish, typically measuring only 6 inches in length, possesses teeth so disproportionately large that they rank among the largest teeth, relative to body size, of any fish in the ocean. The 2 largest fangs in the lower jaw are so enormously elongated, reaching lengths that can equal up to 1/3 of the fish's body length. The fangtooth has evolved special sockets on either side of its brain, channels in its skull, where these teeth can slide when the mouth closes, preventing the fish from impaling its own brain with its weaponry. The fangtooth's appearance is nightmarish by a surface-dwelling standard, with a massive head dominated by huge jaws bristling with needle-sharp teeth, small eyes, and a body that tapers quickly to a narrow tail. Its entire morphology is optimized for a single purpose: to capture and hold onto any prey item it encounters, regardless of that prey's size. Relative to the fangtooth itself, the teeth are not designed for cutting or chewing but for impaling and gripping. Once the fangtooth's jaws close on prey, escape becomes nearly impossible. The teeth interlock like the bars of a cage, creating a prison from which few captured animals can free themselves. This adaptation reflects the harsh reality of life on the abyssal plains. Prey encounters are so rare that a predator cannot afford to be selective about size or a species. If something edible appears, it must be captured and consumed, even if it is nearly as large as the predator itself. The fangtooth spends much of its time in the water column above the abyssal plains rather than on the seafloor itself, engaging in vertical migrations that take it through a range of depths. Young fangtooth fish live in shallower waters and look remarkably different from adults, so different that they were once classified as a separate species. As they mature and descend to greater depths, they undergo dramatic physical changes, their teeth elongating, their bodies darkening, and their morphology shifting to the characteristic adult form. This ontogenetic migration movement to different depths as the fish ages is common among deep-sea species and may reflect changing dietary needs, predation pressures, or reproductive strategy at different life stages. While the fangtooth prowls the waters above the plains, other creatures have adopted a radically different strategy for coping with food scarcity. They simply stay still and wait. The tripod fish, family Bathypteroidae, has evolved one of the most unusual body plans of any fish, transforming 3 of its fins into elongated stilts that allow it to stand on the seafloor like a camera tripod, hence its common name. The 2 pelvic fins and the lower lobe of the caudal fin have been modified into rigid, elongated rays that can extend up to 3 feet in length. far exceeding the length of the fish's actual body. The tripod fish positions itself on these stilts, raising its body above the sediment and facing in the prevailing current, its pectoral fins extended forward and upward at precise angles. The posture is not random but represents an exquisitely refined adaptation for suspension feeding in an environment where actively hunting for prey would expend more energy than the prey would provide. The extended pectoral fins are covered in specialized tactile receptors and possibly chemoreceptors that can detect the minute vibrations and chemical signatures of small crustaceans, worms, and other organisms drifting in the current. When prey contacts or passes very close to these extended fins, the tripod fish can snap them closed, trapping the prey and bringing it to the mouth by facing into the current and maintaining a stationary position. The tripod fish essentially sets a trap and allows the ocean's movements to bring food to it, a strategy that minimizes energy expenditure to levels approaching zero. The fish can remain in this position for hours or potentially days, its metabolism slowed to barely detectable levels, waiting with inexhaustible patience for the current to deliver a meal. The tripod fish's remarkable stilts serve multiple functions beyond simply elevating the fish above the sediment. They also act as sensory probes, detecting the subtle water movements and pressure changes created by nearby organisms. The height they provide gives the fish access to faster-moving currents above the boundary layer, the zone of slower water immediately adjacent to the seafloor, where friction with the bottom reduces flow velocity. By positioning itself in faster currents, the tripod fish increases the volume of water that flows past its sensory fins per unit time, effectively increasing its sampling rate for potential prey. When the fish needs to move, it can simply fold its stilt-like fin and swim, then redeploy them at a new location, selecting spot where current patterns or subtle topographic features might concentrate drifting prey. The waiting game played by the tripod fish and many other abyssal organisms reflects a fundamental truth about life in this environment. Activity is expensive, and in a habitat where your next meal might be weeks or months away, every movement must be justified by potential caloric return. Many abyssal creatures have evolved metabolic rates that are a fraction of those measured in shallow water species, even accounting for the effects of cold temperature. Their cellular machinery operates in slow motion. Their hearts beat at intervals measured in seconds rather than fractions of seconds, and their growth rates are measured in millimeters per year rather than per month. Some abyssal fish are thought to live for decades or even centuries, growing slowly throughout their extended lifespans, reproducing infrequently, and spending the vast majority of their lives in a state of near dormancy. The invertebrate fauna of the Abyssal Plains reveals even more extreme adaptations to food scarcity. Sea cucumbers, soft-bodied echinoderms related to sea stars, and sea urchins are among the most abundant megafauna on Abyssal Plains, sometimes occurring at densities of several individuals per square meter. These creatures crawl across the seafloor on tube feet, ingesting sediment and extracting whatever organic matter it contains, then excreting the processed sediment behind them. They are the earthworms of the abyss, processing and recycling nutrients through their digestive systems, breaking down organic compounds that would otherwise remain locked in the sediment. Some species have evolved elaborate tentacles that they extend above the sediment to capture marine snow before it settles, suspension feeding from the water column, while their bodies remain anchored to the bottom. Other abyssal invertebrates have adopted more active predatory lifestyles despite the energetic costs. Amphipods, small crustaceans related to beach hoppers and freshwater scuds, scavenge across the abyssal plains in search of food. Falls the occasional bonanza of a dead fish, squid, or even whale that sinks to the seafloor. When such a food fall occurs, it triggers a feeding frenzy as creatures from across the surrounding area detect the chemical signature of decaying flesh and converge on the carcass. Amphipods can consume a large fish carcass in a matter of hours, stripping the flesh from the bones with remarkable efficiency. They gorge themselves, storing energy for the long period of famine that will inevitably follow. Studies using baited cameras on the abyssal plains have shown that amphipods can detect food falls from remarkable distances, arriving at bait within hours, even when deployed in areas that appear devoid of life. The Abyssal Plains are also home to numerous species of polychaete worms that burrow through the sediment, creating networks of tunnels that aerate and mix the upper layers. These bioturbators play crucial roles in nutrient cycling, bringing deeper sediment to the surface where its organic content can be accessed by deposit feeders, and transporting oxygen-rich water down into the sediment, where it supports bacterial communities. The worms themselves feed on bacteria, organic particles trapped in the sediment, and occasionally on each other. Their populations can be surprisingly dense, with hundreds or thousands of individuals per square meter of seafloor, though their small size and cryptic lifestyle mean they are rarely seen except when scientists collect sediment cores and examine them under microscopes. Despite the apparent monotony of the abyssal plains, careful examination reveals subtle variations. The seafloor that create local hotspots of diversity and abundance. Small manganese nodules, potato-sized lumps of mineral deposits that form slowly over millions of years, provide hard substrate for attachment in an otherwise soft sediment environment. These nodules are colonized by sponges, anemones, and other filter feeders that cannot survive on the shifting sediment. The areas around nodule fields can support notably higher biodiversity than surrounding plain sediments, creating islands of relative abundance in the desert of the deep. Similarly, the tracks and burrows created by mobile organisms create small-scale topographic complexity that can be exploited by other species seeking shelter or hunting opportunities. The abyssal plains are not entirely lifeless deserts, but neither are they rich ecosystems by the standards of shallow reefs or kelp forests. They represent instead a middle ground, ecosystems running at the absolute minimum energy throughput required to sustain complex life, balanced on a knife's edge between existence and extinction. The biomass per unit area on abyssal plains is typically measured in grams per square meter rather than the kilograms per square meter found in productive shallow habitats. Yet this sparse community processes nutrients, cycles carbon, and supports food webs that connect to the entire ocean through the vertical migrations of organisms and the rain of organic matter from above. Perhaps the most humbling aspect of the abyssal plains is their sheer scale. More than half of Earth's solid surface lies beneath the abyssal ocean, hidden under miles of water, shrouded in absolute darkness. These vast expanses remain largely unmapped at high resolution, unexplored by human eyes, and poorly sampled by scientific instruments. The handful of submersible dives and remotely operated vehicle deployments that have reached the abyssal seafloor represent pinpricks of observation in an area larger than all continents combined. Each expedition discovers new species, sometimes dozens of them, suggesting that the total biodiversity of abyssal plains may rival or exceed that of more celebrated ecosystems. These are species adapted to conditions we can barely replicate in laboratory, possessing biochemistries and physiologies we are only beginning to understand, living lives of patient stillness punctuated by rare moments of feeding, reproduction, and death in the graced wilderness. On Earth, in February of 1977, a team of scientists aboard the research submersible Alvin made a discovery so unexpected, so contrary to prevailing scientific understanding, that it fundamentally transformed our conception of where and how life can exist. Exploring the Galapagos Rift at a depth of approximately 2,500 meters, the researchers were investigating volcanic activity along the seafloor spreading center when their viewports revealed something that should not have existed. In this realm of crushing pressure and absolute darkness, where the nearest sunlight was more than a mile and a half straight up, they encountered not the barren, nearly lifeless landscape they expected, but a thriving oasis, teeming with life in densities that rivaled the richest coral reefs, clustered around fissures in the volcanic rock, where superheated water poured into the frigid ocean. Were communities of organisms, unlike anything in the scientific literature. Giant white clams the size of dinner plates, mussels packed so densely they carpeted the rocks, strange worms in white tubes standing upright like a ghostly forest, and swarms of shrimp and crabs crawling over every surface. The scientists stared in disbelief, struggling to comprehend what they were witnessing, knowing that what they had found would rewrite biology textbooks and challenge fundamental assumptions about the requirement for life. These hydrothermal vents, as they came to be called, form along the mid-ocean ridges, where tectonic plates diverge and magma rises from Earth's mantle to create new oceanic crust. As seawater percolates down through cracks in the fractured volcanic rock, it encounters rocks heated to extreme temperatures by the underlying magma chambers. The water becomes superheated under the immense pressure, reaching temperatures that can exceed 750 degrees Fahrenheit, well above the boiling point of water at surface pressure, but remaining liquid due to the tremendous weight of the ocean above, preventing it from vaporizing. This superheated water becomes highly acidic and chemically aggressive, leaching minerals from the surrounding rocks and becoming enriched with hydrogen sulfide, methane, iron, manganese, copper, zinc, and a host of other dissolved compounds. Eventually, the heated, mineral-laden water finds pathways back to the seafloor, where it erupts in spectacular plumes that billow upward through the cold ocean water like dark smoke. These plumes, often called black smokers when they contain high concentrations of metal sulfides that precipitate upon contact with cold seawater, create chimneys and towers of mineral deposits that can grow to heights of several stories. The structures form as dissolved minerals in the hot vent fluid instantly precipitate when mixing with the near-freezing ambient seawater, building layers of sulfide and other minerals that gradually construct hollow conduits through which the vent fluid continues to flow. Some chimneys grow at remarkable rates, adding inches to their height per day during periods of active flow, while others stand dormant for years before renewed volcanic activity brings them back to life. The largest known hydrothermal chimney structure, discovered at the mid-Atlantic ridge, stood over 160 feet tall before it toppled under its own weight, a cathedral of minerals built by the interaction of Earth's internal heat with the ocean. The chemistry of vent fluids is toxic by surface-dwelling standards, rich in hydrogen sulfide, a compound that gives rotten eggs their characteristic smell and is poisonous to most organisms. The fluids also contain heavy metals, methane, hydrogen gas, and are often strongly acidic or alkaline depending on the rock types involved in heating the water. Yet, it is precisely these toxic chemicals that power the vent ecosystems, providing the energy source that sunlight provides to surface communities. At the base of vent food webs are chemosynthetic bacteria and archaea, microorganisms that have evolved biochemical pathways capable of extracting energy from the chemical bonds in compounds like hydrogen sulfide, methane, and hydrogen. These microbes oxidize the reduced chemicals in vent fluids, using the energy released to fix carbon dioxide into organic molecules through a process fundamentally similar to photosynthesis but powered by chemistry rather than light. This chemosynthesis represents one of the only known pathways for primary productivity that does not ultimately depend on the sun, making hydrothermal vents perhaps the only truly independent ecosystems on Earth's surface. Among the most remarkable inhabitants of hydrothermal vents are the giant tube worms Riftia, which can grow to lengths exceeding 8 feet, making them among the largest colonial invertebrates discovered in the deep sea. These extraordinary organisms possess no mouth, no gut, no digestive system whatsoever, structures that would seem absolutely fundamental to animal life. Instead, their bodies are dominated by an internal organ called a trophisome, a spongy tissue pack, with billions of chemosynthetic bacteria that the worms house in a symbiotic relationship. The worms provide these bacterial symbionts with everything they need: hydrogen sulfide absorbed from the vent water through the worm's bright red plume of gill-like structures, oxygen from seawater, and carbon dioxide from the worm's metabolism. In return, the bacteria use the energy from oxidizing hydrogen sulfide to fix carbon, producing organic compounds that they share with their host, providing all the nutrition the tube worm requires. This relationship is so intimate and so essential that tube worms cannot survive without their bacterial partners. The worms acquire their symbionts early in life, During a brief larval stage, when they still possess a functioning mouth and gut, swimming larvae encounter free-living chemosynthetic bacteria in the water and ingest them. The bacteria take up residence in the developing trophisome, and as the worm matures, the mouth and digestive system are lost, sealed over and reabsorbed, leaving the adult completely dependent on its internal bacterial farm. The tube worm's body becomes essentially a life support system for bacteria, circulating T chemicals and oxygen to the symbionts, and receiving nutrition in return. The red plume that extends from the tube's opening contains hemoglobin, the same oxygen-carrying molecule found in human blood, giving the plume its crimson color. But tube worm hemoglobin has been modified by evolution to transport not only oxygen, but also hydrogen sulfide, binding both molecules simultaneously allowing them to react with each other, a biochemical feat that requires specialized protein structures found nowhere else in the animal kingdom. Tube worm aggregations can reach extraordinary densities, with thousands of individuals packed into small areas around active vents, their white tubes standing upright like the pillars of some alien temple, their red plumes waving in the chemical-rich currents like crimson flowers in an underwater meadow. These aggregations create structural complexity on the otherwise relatively featureless volcanic rock, providing habitat for hundreds of other species. Small snails graze on bacterial films that coat the tubes. Blind shrimp swarm through the plumes. Crab pick their way among the tube bases, and fish hover nearby waiting to snap up unwary invertebrates. The tube worms grow remarkably quickly for deep-sea organisms, with some individuals adding over 3 feet to their length in a single year. Growth rates more typical of shallow water organisms and made possible by the abundant energy provided by their bacterial symbionts. The symbiotic strategy, pioneered by tube worms, has been adopted by many other vent organisms, each with their own variations on the theme. Giant white clams, Calyptogena magnifera, reaching lengths of over a foot, house chemosynthetic bacteria in their gill tissues. These clams position themselves at the interface between cold ambient seawater and the edges of vent flow, where they can access both oxygen and hydrogen sulfide. They extend a specialized foot deep into the sediment to reach pockets of hydrogen sulfide-rich fluid, pumping it up to their gills where their bacterial partners wait. Bathymodialus mussels employ a similar strategy, farming bacteria in their gills while clustering around vent openings where they can filter both food particles from the water and the chemicals their symbionts require. Some mussel species hedge their bets by housing 2 different types of symbiotic bacteria, one that oxidizes sulfide and another that oxidizes methane, allowing them to exploit multiple chemical energy sources depending on local conditions. Among the more bizarre and visually striking vent inhabitants are the yeti crabs, family Cuida, which were unknown to science until their discovery at deep-sea vents in the South Pacific in 2005. These pale, blind crustaceans possess hairy-looking claws and legs covered with dense forests of filamentous, setae bristle-like structures that give them their common name, evoking the mythical abominable snowmen. These setae are not merely decorative but serve as bacterial gardens. The crabs cultivate chemosynthetic bacteria on their hairy appendage, waving their claws through vent flow to provide their bacteria partners with the chemicals they need, then scraping the bacteria off their setae with specialized mouthparts and consuming them. In essence, yeti crabs are farming bacteria, tending their crops on their own bodies and harvesting them for food. Different yeti crab species have been discovered at vents across the global ocean ridge system, from the Pacific to the Atlantic to the Antarctic, each with their own ecological peculiarities, but all employing variations on the bacterial farming strategy. The discovery of hydrothermal vent ecosystem fundamentally changed scientific understanding of life's requirements and possibilities. For decades, biologists had operated under the assumption that all complex ecosystems ultimately depended on photosynthesis, that the sun was the necessary engine powering the biosphere. Even deep-sea communities were thought to rely entirely on the rain of organic matter produced by photosynthetic organisms in sunlit surface waters. Hydrothermal vents proved this assumption wrong. Here were ecosystems where the primary energy source was geological rather than solar. The entire food web was built on a foundation of chemosynthesis rather than photosynthesis. The implications extended far beyond marine biology. If life could thrive in the toxic, superheated, high-pressure environment of hydrothermal vents, powered by chemical energy from Earth's interior, then perhaps life could exist in similar environments elsewhere in the solar system. Jupiter's moon, Europa, and Saturn's moon Enceladus both possess subsurface oceans beneath their ice-covered surfaces, and both show evidence of hydrothermal activity on their ocean floors. These moons receive virtually no sunlight at their surfaces and none in their buried oceans, yet the discovery of vent ecosystems on Earth suggests that life might nonetheless thrive there, powered by the same chemosynthetic processes that support tube worms and yeti crabs in Earth's deep ocean. The ancient Martian surface shows geological evidence of hydrothermal systems, and while those systems are long extinct, they might have provided habitats for early Martian life if it ever arose. Hydrothermal vents thus serve as proof of principle that life does not require sunlight, that biology can find a way wherever chemistry provides an energy gradient to be exploited, that the requirements for life are more flexible and less constraining than we once believed, that organisms inhabiting hydrothermal vents face challenges that seem almost insurmountable. Temperature gradients near active vents are among the steepest found anywhere in nature, with water temperature dropping from over 700 degrees Fahrenheit in the vent fluid to near freezing just inches away. Vent organisms must position themselves in narrow zones where temperature remains within tolerable airable bounds. Too close to the vent and they literally cook; too far away and the chemical concentrations drop below levels that can support their symbionts. Many vent species have evolved sophisticated sensory systems to detect and respond to temperature and chemical gradients, allowing them to maintain optimal positioning even as vent flow rates fluctuate. Some can tolerate brief exposures to surprisingly high temperatures, possessing heat shock proteins and other molecular mechanisms that protect their cells from thermal damage. The impermanence of hydrothermal vents adds another layer of difficulty to life in these oases. Volcanic activity is episodic and unpredictable. A vent that has supported a thriving community for decades can suddenly cease flowing as magma chambers shift or pathways for fluid circulation become blocked. When a vent dies, the community it supported dies with it unless individuals can disperse to other active vents. This impermanence has driven the evolution of remarkable dispersal capabilities in vent organisms. Many species produce enormous numbers of larvae that spend days, weeks, or even months drifting in deep ocean currents, capable of traveling hundreds of miles before settling. These larvae must locate new vents by detecting chemical signatures in the water, follow gradients of hydrogen sulfide or other vent-specific compounds to their sources. The odds of any individual larva successfully locating a suitable vent are vanishingly small. By producing millions of offspring, some vent species ensure that at least a few will survive to colonize new habitat. The study of hydrothermal vent ecosystems continues to yield surprises, even decades after their initial discovery. New vent fields are discovered regularly as deep-sea exploration technology improves and more of the global mid-ocean ridge system is explored. Each new vent field often reveals previously unknown species, some belonging to entirely new families, or even phyla of organisms. The total diversity of vent fauna likely numbers in the thousands of species, many still awaiting discovery and description. Beyond the mid-ocean ridges, hydrothermal activity has been found in other geologic settings in back-arc spreading centers, where tectonic plates converge and one subducts beneath the other, while the overriding plate is stretched and thinned, in submarine volcanoes scattered across the ocean basins, and in unusual off-axis locations where geological structures allow seawater circulation through hot rock. Far from active spreading centers, some of the most recent and exciting vent discoveries involve alkaline vents, also called Lost City-type vents after the Lost City hydrothermal field discovered in the Atlantic Ocean in 2000. These vents differ fundamentally from the black smoker vents discovered in 1977. Rather than being driven by volcanic heat, alkaline vents are powered by chemical reactions between seawater and certain types of rock, particularly a rock called peridotite that originates in Earth's mantle. When seawater reacts with peridotite, it triggers a process called serpentinization that produces heat, alkaline fluids, hydrogen gas, and methane, all without requiring active volcanism. The fluids emerging from alkaline vents are warm rather than superheated, typically below 200 degrees Fahrenheit, and are basic rather than acidic, with pH values that can exceed 11. The towering white carbonate chimneys of Lost City host microbial community that may be metabolically similar to some of the earliest life forms on Earth, making these vents not only oases in today's abyss, but also a potential window into life's ancient origins, proof that wherever energy flows and chemistry permits, life will find a way to extract the energy and build ecosystems from the void. Beyond the abyssal plains, beyond even the deepest hydrothermal vents, the ocean floor plunges further still into narrow, elongated depressions that represent the absolute extremes of depth on our planet. These are the Hadal Trenches, named after Hades, the ancient Greek underworld, and the creatures that inhabit them exist in what is arguably the most physically extreme environment on Earth's surface. The Hadal Zone begins at 6,000 meters depth and extends downward to the deepest point in the global ocean, the Challenger Deep, in the Mariana Trench, which descends to approximately 11,000 meters, or nearly 7 miles, below the surface. At these depths, the physical conditions are so extreme that for decades scientists debated whether life could exist there at all. The pressure in the deepest trenches exceeds 16,000 pounds per square inch, or more than 1,000 atmospheres, roughly equivalent to having 50 jumbo jets stacked on top of every square inch of your body. This is pressure sufficient to compress wood to a fraction of its original volume, to collapse sealed metal containers, and to fundamentally alter the behavior of water itself at the molecular level. The Hadal Trenches are relatively rare features, geological scars carved into the seafloor where tectonic plates collide and one is forced beneath the other in the process known as subduction. There are approximately 33 Hadal Trenches and troughs scattered across the global ocean, most of them ringing the Pacific Ocean in a pattern that traces the Ring of Fire, where the Pacific Plate meets surrounding continental and oceanic plates. Each trench represents a narrow but extremely deep environment, typically only a few kilometers wide but stretching for hundreds or even thousands of kilometers in length. The Mariana Trench alone extends for over 1,500 miles in an arc east of the Mariana Islands, great wound in the ocean floor where the Pacific Plate descends beneath the smaller Philippine Plate. These trenches are, in a very real sense, Earth's final frontier, less explored than the surface of Mars, less studied on the clouds of Venus, places where humans have visited only a handful of times in crude submersibles, at tremendous expense and risk. For much of scientific history, the Hadal Zone was presumed to be effectively lifeless. The combination of extreme pressure, near freezing temperature, complete darkness, and minimal food availability seem to represent an insurmountable set of challenges. Early oceanographic expeditions in the late 19th and early 20th centuries occasionally dredged samples from Hadal depths and recovered a few organisms, but these were generally thought to be accidental captures from shallower water, or dead animals that had sunk from above. The prevailing view held that pressure would eventually reach a level beyond which the fundamental chemistry of life could not function, that there would be a maximum depth beyond which the ocean was truly abiotic. This view began to change in 1960, when Jacques Piccard and Don Walsh descended to the bottom of Challenger Deep in the Bathyscaphe Trieste, becoming the first humans to reach the deepest point on Earth. During their brief time on the bottom, they reported seeing what appeared to be a flatfish swimming past their viewport, an observation that was met with considerable skepticism, but suggested that even the greatest depths might harbor life. It was not until the development of remotely operated vehicles, autonomous landers, and improved camera systems in the late 20th and early 21st centuries that scientists began to systematically explore Hadal Trenches and document their fauna. What they discovered was not a lifeless void but an ecosystem, sparse and strange to be sure, but nonetheless teeming with organisms exquisitely adapted to these extreme conditions. Foremost among the vertebrate inhabitants of the Hadal Zone are the snailfish, family Leparidae, which have proven capable of surviving at depths that would be instantly fatal to nearly any other vertebrate. The current record holder for deepest fish ever recorded is a species of snailfish filmed at a depth of 8,144 meters in the Mariana Trench, nearly 5 miles down. At these depths, the pressure is over 800 atmospheres, yet the snailfish appear completely at ease, swimming gracefully through the water with undulating movement of its gelatinous, tadpole-like body. Hadal snailfish have evolved a suite of remarkable adaptations that allow them to function under these crushing pressures. Their bodies contain no swim bladders, the gas-filled organs that help many shallow water fish control their buoyancy would be instantly compressed to nothing at Hadal depths. Instead, snailfish achieve near-neutral buoyancy through having bodies composed largely of watery, gelatinous tissue that is nearly the same density as seawater, requiring minimal energy to maintain position in the water column. Their bones are poorly mineralized and partially cartilaginous, flexible than rigid, unable to provide the structural support we associate with skeletons, but adequate for an organism that doesn't fight against gravity in the way terrestrial animals must. The snailfish's skull is incomplete, with large gaps that allow pressure to equalize on both sides of the bone, preventing crushing forces from building up. At the molecular level, Hadal snailfish have evolved specialized versions of proteins and enzymes that maintain their function under extreme pressure. Pressure affects the shapes of biological molecules, potentially preventing protein from folding correctly or causing enzymes to lose their catalytic activity. Hadal organisms have countered this through subtle changes in their amino acid sequences that stabilize protein structures under pressure, and through the accumulation of small organic molecules like trimethylamine oxide that act as molecular crowding agents, helping to maintain proper protein function. These biochemical adaptations represent millions of years of evolution fine-tuning metabolism to function in an environment that would be instantly lethal to their shallow water relatives. Interestingly, these same adaptations that allow snailfish to survive at extreme depth also prevent them from surviving at shallow depths. A Hadal snailfish, brought rapidly to the surface, would not explode as popular myth suggests, but its proteins would cease to function properly without the stabilizing effect of pressure, leading to death from metabolic failure. The Hadal snailfish are not passive drifters but active predators, feeding primarily on amphipods, shrimp-like crustaceans, that are among the most abundant and diverse organisms in Hadal Trenches. These amphipods, particularly those in the superfamily Lisianasoidea, have also evolved gigantic forms in the Hadal Zone, with some species reaching lengths of over 1 foot, making them comparable in size to small lobster and earning them descriptions as rat-sized in the popular press. These supergiant amphipods are scavengers, the vultures and hyenas of the Hadal Zone, converging on any organic matter that sinks to these depths. When baited traps or landers carrying fish carcasses or other organic bait are deployed in Hadal Trenches, cameras record the arrival of hundreds or even thousands of amphipods within hours, swarming over the bait in a feeding frenzy that can consume a large fish down to bones in a remarkably short time. The amphipods locate food falls through a combination of keen chemosensory abilities and opportunistic searching behavior. In the food-poor environment of the Hadal Zone, a large food fall represents a bonanza that may not be repeated for months, and natural selection has strongly favored individuals capable of rapidly detecting and exploiting such resources. The amphipods feed voraciously when food is available, storing energy in their tissues to survive the long periods of famine between food falls. Their bodies are packed with lipids at reserves that can be metabolized slowly during times of scarcity. Some species appear to be capable of surviving for months without food, their metabolism throttled down to minimal levels, waiting in a state approaching dormancy until chemical signatures in the water signal the arrival of another meal. The gigantism displayed by Hadal amphipods likely reflects some of the same factors thought to promote deep-sea gigantism, more generally cold temperature, high oxygen availability relative to warm waters, reduced predation pressure, and long lifespans allowing continued growth. In the Hadal zone, these factors may be amplified. The extreme depth and the specialized adaptations required to survive there mean that relatively few predator species have successfully colonized these environments, potentially releasing amphipods from some of the predation pressure that constrains body size in shallower waters. The cold temperatures and high pressures may also slow metabolic rates to such an extent that these amphipods live for many years, possibly decades, providing ample time for growth to large sizes. Beyond snailfish and amphipod, Hadal Trenches harbor a surprising diversity of other organisms. Sea cucumbers have been photographed at Hadal depths, crawling across the trench floors ingesting sediment, just as their abyssal relatives do, extracting whatever organic matter the sediments contain. Polychaete worms burrow through Hadal sediments, their populations dense enough to thoroughly bioturbate the upper sediment layers despite the extreme conditions. Foraminifera single-celled organisms that build shells have been recovered from the deepest trenches, some species apparently endemic to Hadal environments and found nowhere else. Even bacteria and archaea thrive in Hadal sediments, carrying out the same processes of decomposition and nutrient cycling that occur in shallower environments. Their cellular machinery adapted to function under pressure that would destroy most organisms. Each Hadal trench appears to harbor a somewhat different community of organisms, with species composition varying between trenches despite their similar physical conditions. The trenches are essentially isolated habitats, separated from each other by thousands of miles of abyssal plain at shallower depths, for organisms highly adapted to Hadal pressures. The shallower regions between trenches represent barriers to dispersal insurmountable as mountain ranges are to lowland species on land. This isolation has resulted in high levels of endemism, with many Hadal species known from only a single trench system. The Mariana Trench has its own unique assemblage of species, many found nowhere else. The Kermadec Trench off New Zealand has a different fauna, as does the Japan Trench, the Perpetual Trench, and others around the Pacific. This suggests that Hadal life has colonized these extreme environments multiple times independently, with lineages from shallower depths repeatedly adapting to Hadal conditions, and then diversifying within their isolated trench systems. The process of adaptation to Hadal pressures appears to have upper limits. While fish have been documented to nearly 8,200 meters, no fish have ever been confirmed from greater depths, and theoretical calculation based on the biochemistry of osmolite-based pressure adaptation suggests that fish may be unable to survive much below 8,400 meters. At greater pressures, the concentrations of trimethylamine oxide and other stabilizing molecules required to maintain protein function would reach levels that interfere with other cellular processes, creating an insurmountable biochemical conflict. This suggests that the deepest regions of the deepest trenches, from about 8,500 meters to 11,000 meters, may be the domain of invertebrates and microbes only, a fish-free zone representing the absolute limit for vertebrate life. The Hadal zone is also a dumping ground for pollutants from the surface world. Recent studies have detected surprisingly high concentrations of persistent organic pollutants, including banned chemicals like PCBs and flame retardants, in the tissues of Hadal amphipods. These chemicals are transported to the trenches by the same processes that deliver organic matter attached to sinking particles, concentrated in the bodies of animals that feed in contaminated surface waters, and then die and sink, accumulating over decades in the deepest parts of the ocean. The discovery of human-produced pollutants and organisms from the deepest trenches places that have never been directly visited by humans until recent decades, provide sobering evidence of humanity's reach and the interconnectedness of ocean systems. There's no place in the ocean too deep, too remote, or too extreme to escape the chemical fingerprint of human industrial activity. The exploration of Hadal environments continues to reveal surprises. In recent years, scientists have discovered Hadal lakes, pools of dense, sediment-laden water that accumulate in depressions within trenches, creating distinct aquatic environments within the ocean itself. Hadal earthquakes are common, as trenches mark zones of active plate convergence where seismic energy is constantly being released. These earthquakes can trigger massive sediment slumps and turbidity currents that reshape the trench floors and bury whatever community existed there, creating a dynamic environment where catastrophic disturbance is a regular occurrence over geological time. The organisms living in trenches must cope not only with extreme pressure and food scarcity but also with the knowledge, encoded in their evolutionary history, that the ground beneath them is fundamentally unstable. The Hadal Zone challenges our understanding of life's limits and resilience. The organisms thriving in these trenches exist under pressures greater than those found in most of our space, in temperatures near freezing, in complete darkness, with minimal food, in an environment subject to regular catastrophic disturbance. Yet they persist, reproduce, and in some cases flourish, forming functional ecosystems complete with primary consumers, predators, and scavengers. The snailfish swimming through the Mariana Trench at depths exceeding 8,000 meters, the supergiant amphipods swarming over food falls in the Kermadec Trench, the sea cucumbers processing sediment on the floor of the Japan Trench. All these organisms are proof that life's requirements are not as restricted as we once believed, that biology is more flexible and more tenacious than seems possible. They survive in conditions more extreme than most of outer space, in pressures that would crush steel, proving that wherever liquid water exists and energy can be extracted from the environment, life will likely find a way to take hold, to adapt, and to endure. The paradox of the deep ocean in the 21st century is both stark and troubling. We have sent 12 people to walk on the surface of the moon. We have rovers exploring Mars and probes visiting the outer planets. We have telescopes peering into the depths of space to observe galaxies billions of light years away. Yet we have explored less than 5% of our own ocean floor. The vast majority of the deep sea remains unmapped at high resolution, unvisited by human eyes or cameras, and unstudied by science. We know more about the surface topography of Venus, despite its thick clouds and hellish temperature, than we know about the detailed bathymetry of large regions of our own ocean. This ignorance might be excusable if the deep ocean were truly remote and isolated from human influence, a pristine wilderness that could wait for future generations to explore. But that is not the case. Even as we struggle to understand what lives in the deep sea and how these ecosystems function, we are already changing them, already leaving our mark on environments that have remained stable for millions of years. The most immediate and visible threat to deep-sea ecosystems comes from an industry that barely existed a decade ago but is poised to expand dramatically in the coming years. Deep-sea mining. The abyssal plains and sea mounts of the global ocean are carpeted with mineral resources that have accumulated slowly over geological time. Polymetallic nodules, those potato-sized lumps of manganese, iron, nickel, copper, and cobalt that form on the seafloor at rates of mere millimeter per million years, are scattered across vast areas of abyssal plain, particularly in the Clarion-Clipperton zone, between Hawaii and Mexico. Cobalt-rich crusts coat the flanks of seamounts, deposited by seawater over millions of years. Massive sulfide deposits mark extinct hydrothermal vents, rich in copper, zinc, gold, and silver. As demand for these metals increases, driven by the expansion of renewable energy technologies, IC vehicles, and electronic devices, mining companies and nations have turned their attention to the deep-sea deposits as potential sources of raw materials The technology for deep-sea mining is still in development, but the basic approach involves deploying massive machines to the seafloor that would crawl across the bottom, collecting nodules or scraping crusts, then pumping the material to surface vessels for processing. The environmental consequences of such operations are only beginning to be understood, but early studies suggest they could be catastrophic for local ecosystems. The mining machines would destroy everything in their path, crushing organisms and removing the hard substrate that many species depend on for attachment. The sediment plumes created by mining operations would spread for tens or hundreds of miles on deep ocean currents, smothering filter feeders and deposit feeders under blankets of fine particles, potentially taking decades to settle out completely. The noise from mining machinery could interfere with the sensory system of deep-sea organisms adapted to an environment of near silence. Perhaps most troubling, the recovery time for deep-sea ecosystems disturbed by mining would be measured in decades, centuries, or potentially millennia, given the slow growth rates and low recruitment of most deep-sea species. Studies conducted in the 1980s and 1990s, where researchers experimentally disturbed small patches of abyssal seafloor to simulate mining impacts, found that even decades later, the disturbed areas showed limited recovery. The tracks left by experimental mining equipment remained visible. The sediment composition remained altered, and the community of organisms had not returned to pre-disturbance conditions. If these results from small-scale, short-term disturbances are extrapolated to the industrial-scale mining operations currently being planned, the implications are sobering. Mining companies speak of extracting millions of tons of nodules from areas covering thousands of square kilometers. The International Seabed Authority, the UN body responsible for regulating mining in international waters, has issued exploration licenses to more than 20 contractors representing various nations and consortia, allowing them to prospect for minerals across areas totaling more than 1 million square kilometers of seafloor. As of the early 2020s, the transition from exploration to actual mining exploitation has been delayed by technical challenges, economic uncertainties, and growing scientific and public concern about environmental impacts, but the pressure to begin mining continues to build. The deep sea is also become a repository for humanity's waste in ways that would have been unimaginable just a generation ago. Plastic pollution, one of the most visible environmental crises affecting surface oceans and coastlines, has infiltrated the deepest parts of the ocean with alarming thoroughness. Plastic items and fragments have been found in the Mariana Trench, photographed on the seafloor at Hadal depths, recovered from the stomachs of deep-sea organisms, and detected in sediment cores from Abyssal Plains. How does plastic reach these depths? Some sinks directly due to its density or because it becomes fouled with biological growth that increases its weight. Plastic particles are ingested by surface organisms that then die, sink, carrying the plastic to depth in their bodies or fecal pellets. Ocean currents and descending water masses can transport plastic particles to the deep sea. Once there, plastic persists, its degradation slowed by the cold temperature and the absence of UV radiation that helps break down plastics at the surface. The impacts of deep-sea plastic pollution are still being studied, but evidence is mounting that it affects deep-sea organisms in multiple ways. Microplastics have been found in the digestive systems of deep-sea fish, crustaceans, and deposit feeders, having been ingested either directly or through consumption of contaminated prey. These plastic particles can cause physical damage to digestive tissues, block intestines, and potentially leach toxic chemicals including plasticizers, flame retardants, and other additives used in plastic manufacturing. Some of these chemicals are endocrine disruptors that can interfere with reproductive systems even at very low concentration. The discovery of high concentrations of persistent organic pollutants in Hadal Trenches mentioned earlier is likely linked to plastic pollution, as many of the chemicals bind strongly to plastic particles and are transported to depth along with them. In the Hadal Zone, where food is extremely scarce and organisms have evolved to consume almost anything they encounter, plastic particles may be especially likely to be ingested. and to accumulate in food webs. Beyond the tangible impacts of mining and pollution, the deep sea faces a more subtle, but potentially more profound threat from climate change. While the deep ocean might seem isolated from the atmosphere and surface ocean, it is intimately connected to both through ocean circulation, the great conveyor belt of currents that moves water, heat, and dissolved gases throughout the global ocean. The deep ocean is ventilated, supplied with oxygen, and renewed by the formation of deep water. At high latitudes where cold, dense water sinks and spreads along the ocean floor, this process, called thermohaline circulation, operates on timescales of centuries to millennia, meaning that the water in the deepest ocean today may have been at the surface hundreds of thousands of years ago. Climate change is altering this circulation in ways that are difficult to predict in detail, but are likely to be significant. As atmospheric temperatures rise and ice sheets melt, the input of fresh water at high latitudes reduces the salinity, and therefore the density of surface waters, potentially slowing or even shutting down the formation of deep water. Early evidence suggests that some components of the global thermohaline circulation are already weakening, though the magnitude and consequences of this weakening remain subjects of active research and debate. If deep water formation slows substantially, the deep ocean could become more stratified, with reduced mixing between deep and surface waters. This could lead to declining oxygen concentrations in the deep sea, creating more extensive oxygen minimum zones, and potentially pushing some deep-sea organisms beyond their physiological limits. Even modest changes in temperature, oxygen, or current patterns could have dramatic effect on deep-sea ecosystems adapted to stability over millions of years. The deep sea is also absorbing a significant fraction of the excess carbon dioxide that humanity is pumping into the atmosphere, helping to slow the rate of atmospheric CO2 increase, but at a cost. As CO2 dissolves in seawater, it forms carbonic acid, lowering the pH of the ocean in a process called ocean acidification. The surface ocean has already acidified measurably, and this acidification signal is spreading into deeper waters as CO2-rich surface water mixes downward, and as deep water formation incorporates more acidic water into the deep ocean. Many deep-sea organisms have shells or skeletons made of calcium carbonate, a material that becomes harder to produce and easier to dissolve as ocean pH drops. Cold water corals, which form extensive reef structures on sea mounts and continental margins and provide habitat for countless other species, are particularly vulnerable to acidification. Some of these deep-sea coral reefs are thousands of years old, built slowly by coral polyps adding to their skeletons generation after generation. They may not survive the rapid acidification that is projected for the coming decades and centuries. The threats facing the deep ocean are made more urgent and more troubling by our profound ignorance of what exactly we stand to lose. Every expedition to the deep sea discovers new species, often dozens or hundreds of them. Genetic studies reveal that what we thought was a single widespread species is actually a complex of closely related species, each with its own distinct distribution and ecology. The total biodiversity of the deep ocean likely numbers in the millions of species, the vast majority of which remain undescribed and unstudied. Each of these species represents millions of years of evolution, a unique solution to the challenges of deep sea existence, a reservoir of genetic information shaped by natural selection in extreme environments. When we disturb deep-sea ecosystems without understanding them, when we drive species extinct before we even know they exist, we are burning books we have never read, destroying knowledge we may desperately need. The biochemical and genetic adaptations that allow deep-sea organisms to survive in their extreme environment represent a largely untapped resource for biotechnology and medicine. The enzymes from hydrothermal vent bacteria that function at high temperatures have already found applications in research and industry, most notably in the polymerase chain reaction technique that is fundamental to modern molecular biology. and was made practical by the use of heat-stable DNA polymerase from the vent bacterium Thermus aquaticus. Deep-sea organisms produce a remarkable array of bioactive compounds, some of which show promise as antibiotics, anti-cancer agents, or other pharmaceuticals. The mechanisms that protect deep-sea proteins from denaturation under extreme pressure could inspire new approaches to protein stabilization for medical application. The symbiotic relationships between deep-sea animals and chemosynthetic bacteria could provide insight into how to engineer similar relationships for human benefit, perhaps creating new approaches to sustainable food production or waste treatment. Beyond their practical applications, deep-sea ecosystems hold potential answers to some of the most fundamental questions in biology. The discovery of hydrothermal vent ecosystems powered by chemosynthesis rather than photosynthesis demonstrated that life does not require sunlight, a finding that expanded our conception of habitable environment and influenced the search for life beyond Earth. The study of how organisms adapt to extreme pressure, temperature, and chemistry in the deep sea provides insights into the limits of life and the fundamental requirements for biological systems. Some scientists have proposed that life on Earth may have originated at hydrothermal vents, where the chemical energy and mineral catalysts present could have driven the synthesis of complex organic molecules and the emergence of self-replicating systems. If this hypothesis is correct, then studying modern vent ecosystems may provide a window into the conditions under which life began on our planet. Billions of years ago, the deep ocean also plays crucial but often overlooked roles in regulating Earth's climate and biogeochemical cycles. The biological pump, whereby carbon fixed by photosynthesis in surface waters is transported to depth in sinking particles and organisms, sequesters vast amounts of carbon in the deep ocean, removing it from the atmosphere for centuries to millennia. Deep-sea organisms that consume this sinking material and respire it back to dissolved CO2 determine how much carbon remains sequestered and how much eventually returns to the surface and potentially to the atmosphere. Understanding these processes and how they might change in response to climate change and other anthropogenic impacts is essential for predicting future climate trajectories and developing effective mitigation strategies. There are reasons for hope alongside the concern. International awareness of deep-sea conservation issues is growing, driven by improved scientific understanding, compelling imagery from deep-sea expeditions that brings these ecosystems to public attention, and advocacy from environmental organizations. Several nations have established marine protected areas that include deep-sea habitats, restricting fishing and other potentially damaging activity. The international community, through bodies like the International Seabed Authority and Regional Fisheries Management Organization, is grappling with how to balance resource exploitation with ecosystem protection. Though progress is slow and often contentious, some fishing gear modifications and area closures have reduced the impact of bottom trawling on seamount and deep-sea coral ecosystems. The development of less destructive fishing techniques and the establishment of science-based catch limits offer hope that at least some deep-sea fisheries might be managed sustainably, but ultimately, protecting the deep sea will require a fundamental shift in how we value environments that are out of sight and poorly understood. It will require recognizing that the deep ocean, despite being dark, cold, and remote from human experience, is not separate from the rest of the planet but intimately connected to systems that sustain all life, including our own. It will require accepting that our current ignorance of deep-sea ecosystems is not a reason to delay conservation action but rather an argument for proceeding with great caution, for protecting what we do not yet understand rather than exploiting it and hoping for the best. The creatures of the deep sea, from giant squid to Hadal snailfish to bacteria, farming chemosynthesis at hydrothermal vents, have survived and thrived through ice ages and warm periods, through asteroid impacts and volcanic eruptions, through millions of years of environmental change. But they evolved slowly, adapting to changes that occurred over geological time. The changes we are imposing on the deep ocean are occurring over decades, fast than deep-sea organisms can adapt, faster than their ecosystems can adjust. We stand at a critical moment, still largely ignorant of what lies beneath the majority of the ocean's surface, but already holding the power to fundamentally alter or destroy it, and facing a choice about what kind of stewards we will be of Earth's final frontier.